Hi everyone, welcome back to my A-Level Biology lecture series. In this lesson, we will explore limiting factors in photosynthesis. At the end of this lesson, you will be able to state that light intensity, carbon dioxide concentration, and temperature are examples of limiting factors of photosynthesis, and explain the effect of changes in light intensity, carbon dioxide concentration, and temperature on the rate of photosynthesis. Let's get started. Rate of photosynthesis. The rate of photosynthesis describes how quickly photosynthesis occurs in a plant. It can be measured in several ways, including the rate of oxygen release. In this case, we are going to look at the plant, how fast the plant actually produces oxygen during photosynthesis, because oxygen is produced as a byproduct of the photolysis of water during light-dependent reactions. More specifically, is during non-cyclic phosphorylation. So therefore, measuring how fast the plant produces O2 during photosynthesis is a reflection of the activity of photosynthesis. Second, we can actually measure the rate of CO2 uptake, because CO2 is a raw material used in the Calvin cycle for the carbon fixation. The rate of CO2 uptake shows how actively photosynthesis is incorporating carbon dioxide into the organic molecules. Third, we can actually measure the rate of product synthesis, because the speed at which carbohydrates are formed show how efficiently photosynthesis is operating in general. Before we look at the effect of those factors, let us look at the limiting factor and the concept of limiting factors. So when we talk about limiting factor, it refers to an environmental condition that is present in insufficient quantity or in low supply. Therefore, this factor restricts the rate of biological process even when all other factors are in their optimum. By altering the limiting factor will directly affect the rate of photosynthesis. For example, if we increase the value of this limiting factor, we can see that the rate of photosynthesis increases. And increasing it until that factor is no longer limiting, and another factor takes over as the new limiting factor. In this diagram, you can see that this container with a short arm and a long arm. So let us fill in the water, and you can see that the amount or the volume of water that can be filled up in this container or this U-tube depends on the length of the left arm. So if we increase the length of the left arm, you can see that the overall volume of water increases. And this will continue until this left arm now no longer act as limiting factor. Why? Because if we continue to increase the length of the left arm, you will see that there is no further increase in the volume of water. So if we imagine that this is a limiting factor, so when we increase the value of the limiting factor, and the volume of water represents the rate of reaction. So when we increase the value of limiting factor, so therefore the rate of the photosynthesis increases. So from here, we will be able to know that that particular factor is a limiting factor. The reason is because when we increase the particular value, we will be able to see the increase in the overall rate of reaction or rate of photosynthesis until that particular factor no longer act as limiting factor and the other factor take over, or the another factor act as limiting factor. So any further increase of the original factor now no longer increases the rate of reactions or the rate of photosynthesis. However, adjustment to other factor will not further or they will not have any further effect. For example, in this diagram, we can see that a similar tube, or we call it U-tube here, and we fill up the volume. However, when we increase, instead of increasing the first or the left arm, we increase the length of the right arm. And by increasing the length of right arm, does not show any increase in the volume. So this indicates that the factor represented by the right arm is not the limiting factor. So what are the limiting factors in photosynthesis? So the rate of photosynthesis is primarily influenced by three main environmental factors. For example, light intensity, carbon dioxide concentration, and temperature. So how this factor actually affect the stage of photosynthesis directly? For light intensity affect light-dependent reaction, both cyclic and non-cyclic photophosphorylation. Carbon dioxide affect Kelvin cycle directly because carbon dioxide need to be incorporated into Kelvin cycle. And last, temperature affect Kelvin cycle because every single step in Kelvin cycle depends on the enzymes. Even though in light-dependent reaction, we have few enzymes involved, however, the effect of temperature directly affect the Kelvin cycle more than the light-dependent reaction. Let us look at the effect of light intensity on the rate of photosynthesis. Let's look at this graph first. You can see that at low light intensity, the rate of photosynthesis increases when the light intensity increases. And this until the value of K or K light intensity, where any further increase in the light intensity does not increase the rate of photosynthesis, and the rate of photosynthesis is maximum. Above K units of light intensity, there is no increase in the rate of photosynthesis, and this indicates that above K unit of light intensity no longer acts as limiting factor. As contrast to this, below K value or K units of light intensity, the increase of the light intensity increases the rate of photosynthesis, and this indicates that the light intensity is a limiting factor. So at low light intensity, the rate of photosynthesis is low because of insufficient light energy is available to drive the reaction. As light intensity increases, the rate of photosynthesis also increases. And this shows that light intensity acts as limiting factor in these conditions. So with increased light intensity, more light energy is absorbed, leading to the greater photoexcitation of electron from the reaction center. So the rate of non-cyclic photophosphorylation increases, and the rate of oxygen production rises as more photolysis of water occur within the thylakoid space. And there is a greater production of reduced NADP as well as ATP, and both of them are the products of non-cyclic photophosphorylation. The reason why we target or we focus on non-cyclic photophosphorylation rather than a cyclic photophosphorylation, because it totally depends on what we measure. If we measure the rate of oxygen production, therefore, the only types of photophosphorylation that produces oxygen is non-cyclic photophosphorylation. If we want to link it to the Calvin cycle, then we have to talk about the productions of the reduced NADP as well as ATP, because both of them are needed to drive or to make sure that Calvin cycle can be completed. So in the light-independent reaction, where we call it as a Calvin cycle, the reduction of the glycerate 3-phosphate or GP to triose phosphate TP depends on the supply of reduced NADP and ATP. So when light intensity is low, this reduction step becomes the rate limiting step because of insufficient reduced NADP to reduce glycerate 3-phosphate GP to triose phosphate TP. So let's look at this graph again, and you can see that the y-axis indicates relative concentration and the time progress. So at this point, this period of time, there is a decrease in light intensity. So because when the light intensity decreases, what will happen here is insufficient reduced NADP to reduce GP to TP. So you can see that the TP concentration decreases. And why TP concentration of TP decreases but not maintained? Because TP, some of this TP still can be used to form products, and other TP will be used to regenerate back RUBP. And because there is insufficient reduced NADP to reduce GP to TP, so therefore GP concentration increases. And why increases but not stay constant? Because the carbon dioxide fixation step is continued because of CO2 is not a limiting factor. The limiting factor is the light intensity. So overall, you can see that because GP cannot be reduced to TP, GP concentration increases due to two factors. First, GP accumulates because it cannot be converted to TP or reduced to TP. Second, because of the RUBP combined with CO2 to form an unstable 6-carbon compound, which split into 2 molecules of GP. So overall, you can see that RUBP concentration decreases, TP concentration decreases, and also GP concentration increases. The decrease in the TP concentration is due to the use of TP either in the production of the photosynthetic product or the regenerations of RUBP. So as light intensity continues to increase, more reduced NADP and ATP are produced. So therefore, a higher rate of carbon dioxide fixation, and also increase the utilization of CO2 per unit time, and a higher rate of carbon dioxide synthesis as more triose phosphate or TP is converted into glucose and other organic molecules. So when we observe the graph, the y-axis particularly, you can see that if the y-axis, we talk about the how fast CO2 is used, or in the y-axis, we can label it as the how fast the carbohydrates or photosynthetic products can be formed. So at high light intensity, so like for example, above K units, okay, in the graph, the rate of photosynthesis reaches a plateau, remaining constant because another factor has become limiting. So light intensity is no longer the limiting factor. Instead, other factors such as the concentration of CO2, as well as the temperature may limit the rate of photosynthesis. Now, let us look at the effect of carbon dioxide concentration on the rate of photosynthesis. Again, let us look at this graph. The y-axis is the rate of photosynthesis with arbitrary unit, and the x-axis is CO2 concentration arbitrary unit. So again, you can see that when the CO2 concentration increases at low CO2 concentration, the rate of photosynthesis increases. And this indicates that CO2 concentration acts as limiting factor. However, above K units of CO2 concentration, the rate of photosynthesis is constant. Why? Because CO2 concentration no longer act as limiting factor, and other factors such as light density or temperature is too low for the reactions to continue, or for the reactions to increase the rate. So at low carbon dioxide concentration, the rate of photosynthesis increases as CO2 concentration increases. So provided that the factor, light intensity, and temperature are not limiting. And this also shows that CO2 concentration acts as the limiting factor under these conditions. So in these circumstances, the rate limiting step is the fixation of CO2 to ribulose bisphosphate, RUBP, in the Kelvin cycle. As we know that CO2 is needed for the first step of Kelvin cycle, which is carbon dioxide fixation. So let us look at this graph, how we can explain this graph. So first of all, we can see that the concentrations of RUBP increases, and this due to two factors again. The first factor, because insufficient or decrease in CO2 concentration, so CO2 act, or concentration of CO2 act as limiting factor. So without sufficient CO2, RUBP cannot react with CO2, and RUBP concentration should stay constant. However, TP is used to regenerate back RUBP, and RUBP can't react with CO2 because of insufficient CO2. So therefore, RUBP concentration increases. And because lack of the reaction between RUBP and also CO2, therefore GP concentration decreases. Why? Because light intensity is not a limiting factor, and reduced NADP can still be produced by the light-dependent or non-cyclic photophosphorylation. So with the presence of reduced NADP, GP can be reduced to TP. So overall effect here, we can see that GP concentration decreases. And how about TP? And there is no factor that limits the process where TP can regenerate back RUBP, or TP is used for the product synthesis phase. So overall reaction, you can see that TP concentration decreases, GP concentration decreases, but RUBP concentration increases. So as the CO2 concentration increases, the overall rate of Kelvin cycle increases as well. So this enhances the rate of CO2 fixation, so thereby increasing the rate of CO2 uptake, and we can see that the rate of photosynthesis increases. So more CO2 now combines with RUBP to form an unstable 6-carbon intermediate, which is quickly converted to GP. And because of more GP is reduced to form TP, then this allows more carbohydrate products to be synthesized per unit time during the product synthesis phase. And this process also helped to regenerate NADP, which is essential for maintaining non-cyclic photophosphorylation. And be careful in this case, cyclic photophosphorylation does not require an ADP, but with the reduction phase, more GP is reduced to TP, and therefore more NADP can be regenerated for the use in the non-cyclic photophosphorylation. And as a result, there is a continued production of ATP through chemiosmosis and the increase in the O2 release. Why? Because when the rate of non-cyclic photophosphorylation is high, there is more photolysis of water, and therefore there is an increase in the O2 release. So at high CO2 concentration, the rate of photosynthesis eventually reaches a maximum and continue to stay constant. Why? Because another factor now acts as limiting factor. But at this stage, CO2 concentration is not a limiting factor, but other factors such as light density or temperature may limit the photosynthetic rate instead of CO2 concentration. Next, let us look at the effect of temperature on the rate of photosynthesis. So if you look at the graph now, as temperature increases, we can see that the rate of photosynthesis increases. However, this only validates until the K units of temperature, which is the optimum temperature, which gives the maximum rate of photosynthesis. And above K units of temperature, the rate of photosynthesis decreases. And when we observe this graph, we will realize that it is very similar to the effect of temperature on the rate of enzymatic reaction. And how we explain this? We can always explain this by using the idea of an enzyme. So at low temperature, the rate of photosynthesis is low because of the low kinetic energy of the enzyme, such as rubisco, and also the substrate molecules, in this case, RUBP and CO2. As the temperature increases, the rate of photosynthesis generally double for every 10 degrees Celsius increase. This is a principle known as the Q10 effect. So the Q10 effect actually states that for every 10 degrees Celsius increases, 10 degrees Celsius increase in the temperature, the rate of reaction generally will be double all the way, or when the temperature is below optimum temperature. So increasing temperature towards the optimum temperature improves the efficiency of enzyme catalyzed reaction. This is because when the temperature is high, this indicates that the kinetic energy of substrate and enzyme increases. So we can see that RUBP and CO2 as well as rubisco move faster. And therefore, there are more effective collision between CO2, RUBP, and rubisco. And this enhances the CO2 fixation state in the Kelvin cycle. So the rate of CO2 uptake increases. As CO2 uptake increases, the Kelvin cycle proceeds faster. And more GP is reduced to TP, and more NADP is regenerated, supporting a higher rate of non-cyclic photophosphorylation, and therefore increasing O2 production through the photolysis of water. Again, here we focus on non-cyclic photophosphorylation because only non-cyclic photophosphorylation can produce water through photolysis of water. So as TP concentration increases, the rate of carbohydrate synthesis increases as well, resulting in greater products formation per unit time. So overall, when increase the temperature, we can see that there is an increase in the O2 production as well as the products formation and also the rate of CO2 uptake. So at optimum temperature, typically for plant is between 25 degrees Celsius and 45 degrees Celsius, the rate of photosynthesis reaches its maximum. However, beyond the optimum temperature, the rate of photosynthesis declines because some Calvin cycle enzyme, including Rubisco, become denatured. So the active site shape of Rubisco is no longer complementary to the shape of the substrate, RBP and CO2, and prevents the formations of EX complexes. So consequently, the overall rate of Calvin cycle decreases, and this reduces the rate of CO2 uptake, the rate of O2 production. Why? Because less GP can be reduced to TP, and fewer reduced NADP is oxidized back to NADP. And because low concentration of TP, definitely the rate of carbohydrate synthesis decreases. Next, let us look at how we can optimize the photosynthesis in a protected crop cultivation, such as by using glass houses. So an understanding of the effect of environmental factor on the rate of photosynthesis enables farmer to manage this factor more effectively in a protected environment. So, such as by using glass houses with the aim of maximizing crop yields. So the condition inside a glass house can be controlled more precisely compared to the outdoor environments. First of all, we can provide artificial lighting. This can help to extend the duration of photosynthesis beyond natural daylight hours, while also providing a higher than normal light intensity. Second, we can actually provide additional CO2 concentration or increase the CO2 concentration within the glass house atmosphere. By increasing the concentration of CO2, this can help to increase the rate of photosynthesis because we can increase the rate of the Calvin cycle. And third, we can provide extra heating to maintain the temperature close to the optimum temperature for photosynthesis to occur. So previously, traditionally, paraffin lamp have historically been used in glass houses or greenhouses, as their combustion provide not only the light, but also the heat while releases the CO2. And with these three parameters or high value of these three parameters, definitely promoting photosynthesis. And nowadays, the modern glass houses use sensors to monitor the light intensity, the carbon dioxide concentration, temperature, as well as the atmospheric humidity inside the glass houses. Nevertheless, the most important part here, the financial cost of supplying additional light, heat, as well as the CO2 must be weighed against the increased crop yield and resulting income. So whatever we spend or incurred in the cost must be able to generate more income, okay, for the farmer. So ideally, the cost should not exceed the additional profit achieved. No point when we spend more, at the end, the farmer didn't actually earn extra. So farmer must determine the optimum growing condition for each crop by balancing the benefit of increased yield with the cost of maintaining the artificial environment. Other benefits of glass house cultivation include the option to grow plant hydroponically with the roots suspended in the nutrient solution like in the diagram. The ability to vary the nutrient concentration during different growth stages. The use of computerized system to manage environmental condition by detecting those change in the light density, temperature, and readjust back the condition to suit the growth of the plant or the crop plants. And these also improve the control of insect pests and fungal diseases compared to outdoor cultivation, where we may or farmer may have a difficulty to stop the infection, particularly fungal diseases, and also the destroy come from the insect pests. And with that, we have come to the end of this lesson. Hope you found it helpful and easy to follow. If you have any question, feel free to drop them in the comment sections. See you in the next lesson. Have a great day.